Okay, now we're going to look at chapter 15. Chapter 15 is going to look at the microbial mechanisms of pathogenicity. All right, so we saw this chart in the last set of notes in chapter 14. In chapter 14, we looked at the reservoir. We also looked at the transmission. Reservoirs include things like humans, animals, or non-living things like fomites. We also see transmission is through contact. There could be a vehicle like water, food, or air, or vectors like arthropods. Chapter 15 is going to look more at the susceptibility of the host. What's the portal of entry? Did it come through the mucous membrane? Did it come through the skin? That sort of thing. We also want to look at what things that the pathogen has that helps it gain access into the host. How does it also damage the host cells? When we look at how this happens with the damage of the host cells, this is going to lead us into the idea of our immune system, which we'll talk more about in chapter 16 and 17, and also antibiotic therapy, which we'll talk about in chapter 20. We're also going to look at how it damages host cells. Is it due to direct damage? Is it due to toxins? So we have some toxins like exotoxins. These are going to target specific cell types. The body will make antitoxins. We can do preventative things against exotoxins that are called toxoids. This can help with a type of immunization. We can treat this with neutralized exotoxin. With endotoxins, these are going to cause your general aches and fever. This is going to be normally handled with pain relievers. All right, and so we want to be able to see this progression as we go through the susceptibility of the host in order to look at treatments, which we'll talk about in further chapters. So pathogenicity is the ability to cause disease by overcoming the host's defenses. So it's the ability of the organism in order to overwhelm your defenses. Virulence is the degree of the pathogenicity. There are different portals of entry that we are going to look at. These include the mucous membrane, the skin, and what we call parenteral routes. With the mucous membranes, guys, one of the main culprits is the respiratory tract. This is where microbes get inhaled. These can get inhaled via droplets or even dust particles in the air. Some examples that are going to gain access through the mucous membranes will be the pneumococcal pneumoniae, tuberculosis, whooping cough, also known as pertussis, influenza, measles, German measles, infectious mononucleosis, chickenpox, and histoplasmosis. These can all gain access through the respiratory system mucous membrane. Another mucous membrane where they can gain access is through the gastrointestinal tract. This comes from microbes that are in contaminated food, water, your hands, or even fomites. This is known as the fecal oral route because a lot of times these are going to be found in the feces, and if individuals don't wash their hands well and things like that, and things go into the mouth, this can be transmitted. So you have bacillary dysentery, also known as Shigella. We have brucellosis, undulate fever, cholera, salmonella, typhoid fever, hepatitis A, mumps, and trichinosis. These are all just examples of those who can gain access through the membrane of the gastrointestinal tract. Another mucous membrane that potentially is a port of entry is the genitourinary tract. A lot of these you'll notice though, however, are sexually transmitted infections. These include things like gonorrhea, syphilis, non-gonadococcal urethritis, herpes virus infections, AIDS, and candidiasis. If you'll notice, these microbes are transmitted via direct contact, and normally it's sexual contact. The next port of entry is through the skin or the conjunctiva. The conjunctiva is found in the eye. It's the layer in the eye. We see that with the skin, a lot of times there could be broken skin through a cut or an opening. We also see insect bites could be where it penetrates. And then the lining of the eyelids, of course, is the conjunctiva if something gets into the eye. We also have the parenteral. This is going to be where it's deposited under the skin. It's not just through an entry on the skin, but it's under the skin. These are microbes that are deposited under the skin via punctures, bites, cuts, wounds, or types of surgery. Some examples here are gas gangrene, tetanus, Rocky Mountain spotted fever, hepatitis B, rabies, and malaria. And again, these are things we will talk more about in further chapters. So some concepts here, guys. Most pathogens have a preferred portal of entry. They have a way that they would prefer to get into you, whether it's through the respiratory system, the digestive system, through the skin. They may be able to cause disease. They may not be able to cause disease unless they get through that portal of entry. Some pathogens can actually use several portals of entry to enter effectively. They aren't necessarily pinpoint to just one. Virulence varies per organisms. There there must be enough of the organism to gain a foothold to overcome the host's defenses. This would cause the result in a disease. And then almost all pathogens have some means of adherence or attachment. They have some way of being able to attach to your body and your cells in order to gain that stronghold. All right, so penetration of bacterial pathogens. This is known as the invasiveness, the ability of an organism to penetrate into deeper tissue. There are some contributors to this idea of invasiveness. So we talked about some of these when we were looking at the structures of prokaryotic cells. Some of these bacteria have capsules. Capsules prevent them from being phagocyzed, meaning eaten by your immune system. Some of your immune system cells are kind of like Pac-Man. They're able to consume the invader, digest them, and get rid of them. This capsule helps keep it. This capsule makes it harder for those cells to do it, therefore that the bacteria stays around more often. We also see some cell wall components that can actually help with this invasiveness. One is known as the M protein. This is the surface protein. This is associated with strep pyogenes. This promotes attachment and helps resist phagocytosis. We also see some of them may have fimbriae, which helps anchor them into place, those extensions that helps hold them into place. Others have myclonic acid. This is a lipid component. This is going to help things like mycobacteria resist digestion from those phagocytic cells as well. Another contributor to their invasiveness could be the production of exoenzymes. These help organisms to penetrate into deeper tissues as well. An example of this could be coagulases. Coagulases initiate fibrin clots. Staph aureus is one example of this. It's an enzyme that they produce that initiates clotting to take place. We also have kinases. These will actually dissolve clots. So if the body is trying to make clots to kind of fence off the invader, this will actually dissolve those clots. One example is streptokinase, and we actually use this to help dissolve clots when we're talking about somebody having a stroke. Another is hyaluronitis. This breaks down binder proteins, hyaluronic acid, which is found in our tissues. This actually lets the bacteria spread to other areas. We also see collagenase. This is going to break down a structural protein that we call collagen that's in the connective tissue. And we also have IgA proteases. These break down the IgA antibody, which is used by your immune system. We also see that some of them can have what we call antigenic variations or mutations. These are altered surface antigens, which allows organisms from being recognized by your immunity, specifically your adaptive immunity, where your body should have seen it before, and because they have changed just enough, it actually causes it to where your body hadn't seen that before. We also see invasion. These are surface proteins. These are used to disrupt the cytoskeleton in the plasma membrane of a host cell. This actually causes their membrane to start to ruffle, which you can see here in this picture. This ruffling allows them to have greater attachment and greater access to the cell. There's also ways that they can actually damage the host cell. One way is depletion of the host nutrients. There's also direct damage and also toxin production. A quick note here is that most damage is not done by the bacteria itself. It's actually done by the toxins that are potentially released by them. So let's look at toxingenicity. This is the ability to produce toxin. One example is the exotoxin. This is a protein produced by typically gram-positive organisms. This is going to be where the disease is caused by intoxication, not infection. So it's not actually the bacteria that's causing the problem, it's the toxin that they release. This stimulates the production of antibodies. These antibodies, since they're against toxins, are also known as antitoxins. Diphtheria and tetanus are controlled via toxoid vaccines. A toxoid vaccine is where the toxin has been inactivated by either heat or a chemical, and then it gets injected into the individual. This is actually what we have in a tetanus shot. Okay, when you go in and you get a tetanus shot, when they think you might have been exposed to tetanus, this is what's in that shot. And the whole point is it helps your immune system recognize it and fight the toxin off quicker before we actually have the issues. Now guys, these are exotoxins, which means that they are produced by the cell and they are released. They exit the cell, thus the exotoxin. These exotoxins are also soluble in our body fluids and they can spread over long distances. Now, toxins can be grouped according to their action. Neurotoxins are going to affect, of course, the nervous system. Examples of this is botulism. Botulism is going to cause the nerves to not fire to the muscles, making the muscles flaccid or paralyzed. This is going to cause them to not be able to contract. We actually use the toxin that causes botulism in Botox. However, because the bacteria is not present, it only lasts for so long. We also have tetanus. Now, tetanus is the opposite of botulism. Okay, botulism was where the muscles were flaccid and not engaged. Tetanus is where they are contracted and engaged constantly. And so because of this, it causes severe contractions of the muscles. The next one are called cytotoxins. These damage or kill the host cells. Diphtheria is an example. It can actually damage or kill nerve cells, heart cells, and kidney cells. We also have enterotoxins. These act upon the GI tract. They can cause vomiting and diarrhea. Examples of these are cholera, traveler's diarrhea, which is ultimately caused a lot of times by E. coli, and some types of food poisoning. There are also some membrane disrupting toxins. We have the leukocytidins. These kill phagocytic leukocytes or white blood cells, and they also kill tissue macrophages. These are going to be very important factors of your immune system that they're taking out. We have hemolysis as well, which destroy erythrocytes, which are red blood cells. They also can destroy some leukocytes. Example of this is streptolysis. So they can actually get rid of red blood cells and some white blood cells. Now the issue here though is if you don't have a lot of red blood cells circulating, they're not carrying oxygen, so this can cause the individual to also become anemic. All right, so this particular table shows you diseases that are caused by exotoxins. It shows you the disease, it gives you the bacterium, it tells you the type of exotoxin, and it gives you the mechanism. Is it a neurotoxin, a cytotoxin, an enterotoxin, so on. You will notice though, guys, I marked on here that most of these are gram-positive bacteria because most of them are going to be creating this exotoxin because they are gram-positive. There are a few exceptions, with a few being gram-negative. The next are endotoxins. Endotoxins are associated with lipid A of LPS in gram-negative organisms. All right, so exotoxins were bigger in gram-positive. Endotoxins are more of an issue in gram-negatives. These are usually less toxic than exotoxins. All endotoxins produce the same signs and symptoms. They cause general chills, fevers, and aches. All right, so when we talk about having those like chills and aches and pains from being sick, these could be caused by endotoxins. Some are associated with causing DIC. DIC can be a life-threatening condition where the body has clotted too much and used all its clotting factors, which means then they're bleeding somewhere else. All right, so it's kind of a vicious cycle of trying to figure out and keep clotting and bleeding under control, and it could also lead to septic shock. Endotoxins are not very antigenic, meaning antibodies are not usually produced against these guys, and so there's no vaccines available. Now, when we look at endotoxins, these toxins are found inside the cells. The only time these toxins leave the cell is when the cell has been destroyed, when the bacteria dies. So this is almost kind of like its last hurrah and saying, hey, you killed me, but here's my toxin, and it's still going to cause some damage. It's still going to cause chills, fevers, and aches and pains. All right, so this particular chart takes exotoxins and endotoxins and it does a comparison with them. It shows you that exotoxins are mostly from gram-positive bacteria, whereas endotoxins are more gram-negative. It tells you a little bit about their chemistry, their pharmacology. Are they heat stable? Are they very toxic or not? So exotoxins are highly toxic, endotoxins not as much. Do they cause fever? Exotoxins do not, endotoxins do, and the list kind of goes on and on. We also see for exotoxins, it shows you the representative diseases. These are things like gas gangrene, tetanus, botulism, diphtheria, and scarlet fever. For endotoxins, these are produced that cause typhoid fever, urinary tract infections, and meningococcal meningitis. All right, so when the balance between host and microbes gets tipped in favor of the microbe side, an infection or a disease can result. Learning these mechanisms of microbial pathogenicity is fundamental in understanding how these pathogens are able to overcome the host defense. How are they able to overcome it in order to make you sick? All right, because if they weren't able to do that, then you just wouldn't be sick with anything. And so when we look here, the portals of entry are important, but also, do they have a way to penetrate or evade the host defenses? Do they have a capsule? Do they have those special cell wall components? Do they have enzymes? what kind of damage to the cells do they do? Or is it due to toxins? What is the portal of exit? How do they leave and get and transmit to somebody else? These are all things that are really important when we are discussing how these microbes gain an advantage over our immune system, but also how they're passed from one person to the next. All right, so that was the focus of chapter 15. Again, if you have any questions or concerns, please let us know.